very nice introduction, not dry at all, so thank you for that. I appreciate that. Before I start, I should thank certainly Dr. Hamilton for inviting me here to speak. I'm grateful to the Health and Exercise Science Department, as well as the Columbine Health System Center for Healthy Aging for sponsoring my trip, so I have to say that. Today is Good Friday for those of you that are in the Christian world. It's Passover tonight for those of you in the Judeo side of life. And with that combination, I think that's the answer for any of your struggles. I'm sorry about that. But I just want to thank everyone for coming. Super excited that you're here, because I am super excited to talk about a project that is really totally new. It's a new direction for my lab. And so for students out there, the message is you can rise to whatever level you want to, but the great thing about academia is you can start over again. So I have a long history in nutrition-related research, amino acid metabolism, protein balance, and I still do that. But this was an area that I had an opportunity to step into, and I'm hoping it will continue. I have a great collaborator in the Equine Science Center at Rutgers, and I should point that out as well as who did the work. So before I tell you about this, let me tell you who did the work. So what I'm going to talk about today, a lot of it, almost all of it is unpublished. It's right now in that peer review world, okay? So you'll see it soon, hopefully. And it is the hard work and efforts of a talented PhD student in my lab, Dylan Klein. So you'll see here, he is on the far left, that good-looking gentleman in the blue shirt. And so he got his PhD last year and is now faculty at Rowan University in New Jersey. And the rest of my lab helped him out tremendously. So Emily Merrick was the woman in blue in the middle, the top picture. She's my lab manager. And Young Ping is right next to her. So these three were kind of the trifecta of the data that I'm going to show you. And my co-investigator on the work is Ken McKeever at Rutgers University. He's the horseman. And I know he has done a long history of work in Lasix and horse performance. So he'd be the one to answer any of your questions in equines. I'll do my best, but I will tip my hat to him and others at the Equine Science Center for providing this really cool project. So let me start by giving a brief overview of what I'm going to tell you today. So first I'm going to start with some description of some molecular underpinnings of exercise as we know it as medicine. And I'll give a broad overview, but I want to focus in on a pathway that my lab has spent many years studying, and it's called the unfolded protein response. And so this is a pathway that has a lot of data as with respect to nutrition. Less is known with exercise, and certainly nothing was known in the horse. So it's really a totally new area. So I'm going to introduce this pathway to you for those of you that aren't familiar with the unfolded protein response, and to talk about how an aspect of that is called the integrated stress response. So I kind of use these terms. I go back and forth between them. And so with that introduction, I'll move into this project of equine responses to acute exercise and also to training in terms of going from the unfit to the highly fit state, and how acute responses to exercise are similar versus different in the equine species. And along with sort of looking at molecular underpinnings, I also looked at metabolism. So we looked at the skeletal muscle metabolome in these glorious creatures. So I'm going to share with you some of the findings that we found there. And then I'll summarize. So this is a figure taken from a very famous exercise physiologist, Julene Zaref. She recently received the Burson Award at Experimental Biology from the American Physiological Society. And I think that this figure from her review, that's 5 years now ago, but it gives an illustration of what I think is true for those of us that study exercise. When we get into studying exercise, we have to make a choice. Are we going to study something that happens right after we work out? There are these acute exercise responses. Or are we going to study what happens after adaptation to repeated doses of exercise. And what we find is that acutely, any one of these events, these bouts of exercise, can cause wide shifts in certain kinds of measures, measures at the level of signaling, measures at the level of mRNA. And every one of these responses appears necessary, but none are sufficient by themselves to result in adaptation and improvement of fitness. So it is this repeated dosing over time that accumulates and results in increases in performance. So it's a nice illustration here that you can see that early on you get these peaks, but you're not going to see improvements in exercise performance or whole muscle metabolism. You have to take it over time. And when you get to the point of seeing changes in performance and metabolism, these acute responses don't always occur anymore. So I find that's a model, and I bring that up because I think that is essentially kind of the story that I'm going to tell you as it relates to the horse. So this is sort of a messy slide, but it kind of gives an appreciation for how complicated these acute events are when it comes to intracellular signaling. So in mammals, there are at least 3 nodes, central nodes of signaling that we know are important for exercise responses and regulating protein balance. So on the right, you've got the growth factor, AKT pathway, which can regulate both synthesis and degradation. Central there, you have mTOR signaling, which we know integrates a lot of different information from the environment and can influence protein synthesis and degradation. And then we have on the left the whether it's through calmodulin kinase or AMP kinase LKB, you've got that AMP kinase PGC1 alpha signaling pathway, which is really important in mitochondrial biogenesis and remodeling. So whether you're on the aerobic side or you're on the resistance side, or maybe somewhere in between, you've got all of these pathways being engaged. And it's sort of the challenge for those of us that study exercise responses. Which one of these are necessary to be able to improve fitness? So a lot of people spend time on these signaling pathways, and what I have noticed is that not a lot of attention was being given to the one that I like to study with nutrition, and that's the unfolded protein response. So let me tell you a little bit about the unfolded protein response. So in the skeletal muscle, we have a very important organelle called the sarcoplasmic reticulum. And in any other tissue, we call it the endoplasmic reticulum. And this is a space in which proteins are made that are destined to be secreted or become membrane proteins. Now, in the muscle, it's also a very important place where calcium is sequestered, and it regulates the release of calcium into the cytosol. And calcium is very powerful. It creates a very important mechanotransduction signal for the muscle. And so it is really important that the environment of the ER is regulated in terms of homeostasis. And so there are a number of signaling molecules that are in the membrane of the ER, or the sarcoplasmic reticulum, and these signal transduction proteins monitor the environment of the ER. And the aim of these proteins is when they sense that the environment is disrupted, when there's no longer homeostasis, it performs 3 major responses. So if there's either an accumulation of misfolded proteins or calcium balance is disrupted, the first response is to reduce global protein synthesis. So reduce the client load of what's being made in the ER. It's very energy expensive, very taxing. And if you've built up a lot of stuff in the ER that really shouldn't be there, that's kind of a signal for cell death. So reduce global protein synthesis. Second response is to actually help refold any misfolded or unfolded proteins. So you have a lot of chaperones that come in, and they just help monitor and manage anything that's not being folded properly. The third is any trash that's in there that can't be refolded needs to be thrown out. And so this is through a process of ER-associated degradation. So
So you think about like an assembly line. Proteins are getting made, and if something happens in that assembly line, you have to be able to first turn off the assembly line to pick up the mess and throw out the trash. And that's essentially what the ER does. Okay, that's the unfolded protein response. So those are the main three kind of functional events. And to achieve that, there's going to be, both at the level of mRNA and the level of protein, there's going to be changes. So the transcriptome will be altered, and also genes will be altered at the level of protein synthesis in a very specific way. So this unfolded protein response is associated with all sorts of diseases. And, you know, it's kind of a challenge when you study this. Does this response cause problems, or is it actually a means to prevent an even worse outcome? And I would say, by and large, the unfolded protein response is there to be protective. It's actually there to prevent an even worse outcome. So we like to think of it as pro-adaptive. It gets acutely activated in order to be able to protect the cell against future stressors, to be able to build up resilience. So if it's acutely activated and it resolves in a nice way, that's good. That's a productive ER stress response, or UPR. But if the environment of the cell is such that these stresses cannot be remediated, then there's continuous activation. And so it becomes kind of hyperreactive. And in that case, that's when it becomes maladaptive. And chronic cases of ER stress are what is associated with everything from Alzheimer's to actually sarcopenia and skeletal muscle atrophy. So in a broad way, that's kind of what I like to study, is what is it about the UPR that is making a cell more resilient to stress? And at what point is the tipping point to make the UPR going from being productive to being maladaptive? So let me tell you a little more detail. So these three arms, because when I show you the data, you're going to have an appreciation for what I'm showing you. The three arms are, the first is what we call the integrated stress response. Starts with a kinase called PERK. And this PKR-like ER resident kinase. PERK is in a family of 4 kinases. And PERK is sensitive to ER stress. When it gets phosphorylated, it will then phosphorylate a translation factor called EIF2, eukaryotic initiation factor 2. Very important in the initiation step of protein synthesis. So PERK regulates protein synthesis globally, kind of slows it down. That's that first important step, right? Slow protein synthesis. And in slowing protein synthesis, it actually causes certain mRNAs to be preferentially translated. So on a background of most mRNAs not getting made, a certain class are getting made more frequently. And the best characterized is one called ATF4, shown here. So ATF4 is essentially a transcription factor. There you go. It's a transcription factor. It's a basic leucine zipper transcription factor. And it has binding partners like NRF2, Dr. Hamilton's favorite protein, and will realign the transcriptome in a way to promote resilience and protection. And this pathway is what I study a lot when it comes to amino acid stress responses. The second is called ATF6. So ATF6 is activated a little differently. When there is unfolded proteins, chaperones go to refold, and that leaves ATF6 kind of naked without its binding partner. That causes ATF6 to be released from the ER, moves to the Golgi, and kind of like SREBP, it gets cleaved by proteases in the Golgi. And so then the cleaved portion of ATF6 can then be a powerful transcription factor. Now ATF6, there's a body of work that says that PGC1 alpha is also involved in binding ATF6, and so that's part of that exercise response, is through ATF6. Finally, IIIY alpha, it's a kinase, but it's also an endoribonuclease. It's targeted toward this XBP1 mRNA, and when XBP1 gets cleaved internally, it becomes an active transcription factor. So these are the three arms of the UPR that collectively function to be protective. I mentioned that not a lot of information is known about the UPR in exercise, and almost nothing in horses. But let me tell you what is known. So I'll give you a little overview of what's out there when it comes to humans or rodents with respect to the UPR and exercise. So one of the very first studies was by Bruce Spiegelman's group, and he basically showed that after a single bout of exhaustive treadmill exercise, and this is in mice, and essentially you see that there is an increase in the phosphorylation of EIF2 in the muscle, and then this chaperone, so this would be a chaperone in the ER that helps refold misfolded proteins, that is also induced in the ER muscle. So it looks as if there is at least two arms of the unfolded protein response activated by exercise. And then down here you can see that these same markers, but a whole slew of what we call biomarkers of the UPR, they are all increased with that single bout of exhaustive exercise. But if you take those mice and then train them to be highly fit, and then redo this study, no longer do you see this increase in any of these markers. It's either significantly less or no increase at all. So this gives you a sense that this UPR happens under unaccustomed exercise. And when you become accustomed to it, you do not see the same response. If we look at humans, the first study that examined UPR in humans used subjects that were engaged in a 200-kilometer ultra-endurance run. And 3 hours after that run was complete, somehow they got these subjects to give a muscle biopsy. Okay, so 3 hours after this 200-kilometer run, they got a muscle biopsy, and what was seen is that XBP1 and ATF4 were both increased in the skeletal muscle 3 hours after that ultra-endurance run. So both cases, endurance exercise, a few hours after the cessation of that exercise, you see the UPR up. Now, let's compare that to resistance exercise. So this is a human study, and it followed subjects for 3 hours, 24 hours, and 48 hours after a single bout of resistance exercise in those that were unaccustomed to that workout. And what was seen is that in all cases, that on the left here we have splicing of XBP1, and what you can see is that there is an increase in XBP1 splicing, but it doesn't rise significantly until 24 to 48 hours after the bout. So there's a delay. So endurance exercise pretty early, resistance exercise later. And that was true for any of the markers that they looked at, that essentially it took 24 to 48 hours in order for the UPR, any marker they looked at, to see an increase. And then finally, here's another study I wanted to point out. Again, this is a human study looking at unaccustomed resistance exercise. And it pretty much agrees with the previous study in that the UPR is induced in the skeletal muscle, but at 24 to 48 hours. So, this sort of let us know, when we were looking at a design, we were trying to figure out in horses, what is the best time point to pick? So we have this endurance exercise about 3 hours, and we have our resistance exercise models that are 24 to 48 hours later. So if I can summarize kind of the state of the union, if you will, as we went into this study, is that the mammalian literature tells us that ER stress and the induction of the ISR and the UPR was really seen with unaccustomed exercise. If you had a trained individual or trained rodent, we really didn't see induction. And so, how much that these responses directly mediated adaptations, we don't know. So let me move now to the idea of moving to the horse. Why would we do that? Well, the horse is exceptionally muscular for its body weight, over 55% skeletal muscle mass, and they're natural athletes.
and they really depend on normal skeletal muscle function for health and performance. We know that there really is not good information at the level of molecular control, but in New Jersey, they're our state animal, believe it or not. They're on our flag. And the equine industry in New Jersey is valued at $4 billion. It generates over $1 billion annually in New Jersey, and about 20% of our agricultural acres in New Jersey are equine related. That may surprise some of you in Colorado when we're a small state, but the horse is important to New Jersey. So there is a number of investment parties in New Jersey that want to know how to aid in exercise and training, and what are the best strategies to improve health and performance in the animal. What I can tell you, I didn't know this until getting into this field. The horse is the only other mammal that thermoregulates by sweating, like us. I thought that was a fascinating angle for those of you that don't know that. And so, they are a unique model for exercise responses because they are like us in that way. And, if you're interested in novel models for aging, I think the horse might provide a really interesting path forward. They certainly have plenty of muscle to donate to the process without ever having to get rid of them. They're an interesting model to sample and utilize over and over. And their lifespan is long, so they're a nice model, I think, for humans. So this is what we came up with. We have two hypotheses that I'm going to provide some data to address. So the first hypothesis is that acute high-intensity exercise that will result in volitional fatigue will stimulate the ISR, stimulate the UPR, and we'll see that in equine skeletal muscle post-exercise in the untrained state. And on the other hand, we'll find that by training them, maybe there'll be differences basally in the UPR, but we might find that the responsiveness, like we see in the rodent and the human literature, the responsiveness will disappear as they become highly fit. And so we're seeing these markers as a way to show whether or not the animal is fit or not. So this is our overall experimental design. So this study is unique in that the horses were served as their own control. It was a crossover design, 8 horses, 4 mares, 4 geldings, between the ages of 3 and 8 years old. They were standard bred horses. They were housed by sex in groups of 4 in their respective 2-acre paddocks leading into the study. And you can see that at the top, we have an exercise EX group, and we have a standing control SC group. So we brought in these horses that were considered to be unfit, and I'll go into more detail about that in a little bit. And the way we would design the study is we had 2 mares and 2 geldings that were assigned to the EX group, same assigned to the SC group, and then they conducted this single bout of a graded exercise test. So we got them to a point of volitional fatigue. We then put them back in their paddocks and gave a 2-week washout, and then the 4 that were the standing controls became the exercise group, and the 4 that were the exercise group now became the standing controls. And so before and after the exercise, we collected blood, we collected skeletal muscle biopsies. So let me show you here, I have a video, if I can find my, okay, so this is our horse treadmill. This is Dylan taking a blood sample from a horse. And so we would start the horse at 4 meters per second for 1 minute. After that 1 minute, we would increase to 6 meters per second for 1 minute. And then every minute thereafter, we would increase by 1 meter per second until the horse would come to volitional fatigue. And we consider volitional fatigue the point at which natural encouragement by their trainers and owners was failing to keep them going. So during what we call the GTX trial,
Oxygen consumption and carbon dioxide production was measured every 10 seconds. We had an open flow indirect calorimeter that was on the horse. And blood was collected during the final 10 seconds of each incremental step to measure lactate. And about 20 mils of blood was collected at each time point. So here's sort of the outline of what was collected, and you can see that right before the exercise, we took a biopsy, and then we took a serial blood sample during the graded exercise test, and then we took muscle biopsies at 3 hours and 24 hours. And so I provided you that literature before, so you know why we chose the time points that we chose. We decided we wanted to be able to understand, does this graded exercise test, is it more like an endurance run, where 3 hours we would see changes, or is it more like a resistance exercise because it's such an acute to volitional fatigue that we might not see something until 24 hours later? And so we did this in the exercise animals as well as the standing controls. And we took our muscle biopsies using a percutaneous needle, and it was obtained from the gluteus medius with suction, and following a lidocaine injection, and we made sure that the subsequent biopsies were at least 2 or 3 centimeters away from the initial incision. Same incision, just the needle was moved away to be in a different area of the horse. Now we know that what we sampled, for those of you that are muscle people, we know that we sampled essentially an area that would be type 1 fibers. Okay, so that's the acute exercise in the unfit, untrained horse. And then we started training them. So that began in April and was concluded in July of the same year. So we had 12 weeks of conditioned exercise that was carried out 4 days a week using an equicizer, which I'll show you a video of in a little bit. 4 days a week on the equicizer, 1 day a week on that same high-speed treadmill that I just showed you. So after that 12 weeks of training, we gave the horses a 72-hour washout period, and then we conducted the same GTX that I just took you through. So we had one group that was an exercise for horses, another 4 that were the standing controls, and then a 2-week washout again, where they would switch groups. And during that washout, they would continue on the equicizer, so we maintained their fitness in between. So let me show you the equicizer. So this is our equicizer at Rutgers. And you can see that on the weekends they were in their paddocks. On Mondays, Wednesdays, and Thursdays, they had what was considered a light sort of workout, whereas Tuesdays was considered moderate, where they would get up to a trot or a slow gallop, and then Fridays were always on that graded exercise test on the treadmill when we get them to fatigue. So that was sort of the plan for the 12-week period. Okay, so how fit did we get them? So let me tell you, for those of you that aren't into different species, right? So we talk about aerobic capacity, right, as a measure of cardiorespiratory fitness, right? So let's start with Homer, okay? So Homer would be just above 20 mils per mg per kg, okay, in terms of their VO2 max or their aerobic capacity. But you know, probably anyone here in Colorado, you'd be somewhere more in the 40 to 50 range if you're a male, okay? And if you were to get trained, maybe already you're up in the 60s, okay, range. Because that's a pretty fit male. Now, if you really want to talk exercise fitness though, then you have to look, certainly you've got to look to, you know, the cross-country skiers, because they're the best in the world. You know, but let me give you a name. So Lance Armstrong, you know, he was measured 85. That's pretty good. That's very good, okay? So what's the world record? So there's a controversy. You got Bjorn Daly in 2001, and he was 96.
And then Oscar Steffensen was 97.5, both Norwegian cross-country skiers. So they're both not quite 100. So the best human in the world is not quite the greyhound, okay? So the greyhound dogs are even better in terms of their exercise capacity, but that doesn't even start to describe the horse, okay? So the unfit horse, the really lazy Homer Simpson of horses, is going to be around 120, all right? Now if you want to talk getting really fit, so you get into thoroughbreds or you get into the high performance racers, and now you're up around 180 in terms of aerobic capacity. Now that's not the best. Sled dog huskies have been recorded up to 240, right? Okay, so you get a sense of what the range might be, and the scale that we're working in when we start to work with the horse, okay? So where did our horses fall at Rutgers? So here's sort of a broad grouping, combining the mares and the geldings, and I can find you can see the GT, the VO2 max, mils per kg per min, 148 is where they started out before training. So they're not the most unfit horses, but I think what was encouraging is those 12 weeks of training got them up to 162 on average. So we definitely significantly improved their exercise capacity as a group. And in that, you could also see that the VO2 max improved, the max velocity, so meters per second went from 9 to almost up to 11. And so the total run time improved significantly, as well as the total distance. So we definitely made them more fit. If we want to break that out in terms of sex, because we had that capacity, having 4 and 4, what we found is that initially before training, there was very little differences, at least statistically, between the sexes. There was, by your eye, if you really discriminately look at the numbers, you'd say that the geldings, tended to have a little more fat-free mass, a little less body fat, a little greater VO2 max. The only thing that came out statistically significant was the total run time. But after training, we did see some sex differences. So I would say in general, the geldings were leaner. They lost body fat during the training, where the mares really didn't. Mares maintained their body fat. And so certainly any exercise physiologist would say when you're performing an endurance run, the less body fat, the better performance. So, and I think you kind of see that here, where the mares had a little more body fat, but they also had a little less performance statistically compared to the males. But they still improved tremendously and statistically from their before training period. So here's the differences that we see in the performance, the VO2 max, as well as the max velocity, run time, and distance. Okay. So did we get them to fatigue? Did we get them at a real exertion? And yes, we did. So if you look at plasma lactate, you can see that both before training and after, plasma lactate increased significantly. And in fact, you can see that it's even higher in the after training. So it's illustrating the high intensity and anaerobic nature of the exercise bout. One of the validated markers, in response to exercise has been PGC1 alpha. And so this is the mRNA expression of that marker. And what we saw is similar to the rodent literature, similar to the human literature, in horses we see an increase in mRNA expression of PGC1 alpha after 3 hours after exercise, but not 24 hours. And this is the unaccustomed state. So now I want to share with you what we found in the UPR. So this is the first data that we know of in the horse that looked at the UPR. So let's start with the phosphorylation of VI2 alpha. So we would predict that in the unaccustomed exercise state, that we would see an increase in the phosphorylation of VI2 alpha after exercise. And in fact, that is what we saw. So we see a significant increase in phospho-EIF2 in the muscle of horses in the untrained state. However, after training, when they were highly fit, that increase disappeared. So there was no longer a significant increase in phospho-EIF2 in the muscle of the trained horses.
So let's move down that path. We're still in the integrated stress response. Phospho-EIF2, when you see an increase in phospho-EIF2, you should see more ATF4. And you can look at ATF4 at the protein level, but you can also look at mRNA levels, because ATF4 protein will actually make more of its own mRNA. And antibodies are not as good and don't have as many in the equine species. So we designed primers and looked at mRNA levels of ATF4. Okay, so if we look at ATF4, what we found was that there was a trend. It didn't reach, it was between 0.05 and 0.1. There was an increase in ATF4 mRNA at the 3-hour, not statistically significant at 24 hours after exercise. And this is, again, unfit, unaccustomed state. If we look at BIP, which is a chaperone, when these proteins that help refold in the ER, we did not see any change in that over time. So, in terms of the unaccustomed state, we saw evidence that the UPR has some evidence of being active early on. So when it comes to training, we really didn't see a lot of changes in the UPR in the trained state. But what we did see is we found that many of these markers actually reduced in expression basally. So there's kind of 2 points to make here. One, in general, if you notice the gray bars are generally lower than the black bars. So the expression overall of these stress markers are reduced in the muscle basally of fit horses. The other notable feature to point out is that it was the variance that we see really matters. So in our unfit horses, great variance in the responses in many of these markers. But as they became highly fit, they became much more like each other. The variance was compressed. And so basically we saw that this became a pattern that we started to see again and again. The other pattern we saw was, so this is the integrated stress response, and one of the targets of ATF4 is this transcription factor called GAD45A. So, this ATF4 GAD45A axis has been looked at by someone who I admire in the field called Chris Adams. He's at the University of Iowa. And he has a whole research program that's in rodents, but what he's been able to discover is that ATF4, when it's overexpressed, at least in mice, it causes muscle wasting, and it's through this GAD45A. And he's found some small molecule inhibitors that if you inhibit ATF4 with these inhibitors, you actually make the muscle resistant to sarcopenia and to loss by fasting and hind limb suspension. So we wanted to know, how does this axis respond to this form of exercise and training? So as I showed before on the left, ATF4 mRNA is increased at 3 hours after exercise in the untrained state. But as the animal becomes trained, that disappears. And if you look at GAD45A, actually you find that wide variation, but kind of a higher level of expression in the untrained. And when the animals become trained, it's a lower and tighter level of expression. So for us, that kind of says what exercise is doing is down-regulating a path towards sarcopenia. The other thing that we saw with our training paradigm was expression of E3 ligases. So as I mentioned, part of the unfolded protein response has to do with ER-associated degradations, taking out the trash. And these two markers are essentially kind of the tags that tag ubiquity on these proteins that need to be shipped out. And we found that with training, both of these biomarkers increased in the muscle of trained horses. So we think this has to do with proteostasis control, and in fact, it's a better, more efficient way to manage muscle to have an increase in expression of these proteins. So let me summarize this for you. So we performed an acute fatiguing exercise in the untrained state, and we saw an increase in the IF2 phosphorylation and sort of that activation of the integrated stress response. Then we made them highly fit. When we looked again we no longer saw that E-active phosphorylation was increased, and even though the animals were, having a greater amount of work being completed, and that chronic training reduced basal expression of many of these biomarkers, and we saw compressed variability. Whereas markers of the proteasome degradation, we saw that increased. So what can we take home? What I can take from this is that the UPR is a pathway that coordinates adaptive responses to stress, and that intermittent activation is helpful for adaptive responses. What we don't want to see is sustained activation. In many models, that's maladaptive. So that's why it seems to make sense that unaccustomed exercise across species activates the UPR, whereas the trained state of that response disappears because exercise training habituates the UPR. And that when you see that disappearance of the UPR, that's going to correspond with improvements in performance and improvements in metabolism. So now the last part of this talk is I want to talk about when we got this UPR story. We wanted to say, well, we noticed that there was this compression of variance in the fit state, and we didn't see a lot going on in the UPR. So what happens to metabolism? What happens to the muscle metabolism? And if you look at the data, at the literature, you'll see that the horse is the most ignored species when it comes to large agricultural animals. There's this great resource of metabolomics available, and it's hardly been utilized in the horse. So we thought that this was a really interesting path to go down to understand what happens in the muscle of horses. So what happened? So what we did, we had a relationship with metabolon, and so we had these muscle biopsies that we were using for the UPR. And so we used a subsection of these muscle biopsies and sent them to metabolon. And they essentially used ultra high performance lipid chromatography, tandem mass spec analyses. They have their own platform, and they were able to deliver to us a discovery panel of metabolites. And so then we've taken that, and what I'm showing you here is something called a principal component analysis. It's essentially a way to kind of, in a big data sense, tell you which samples cluster like each other. It's kind of an easy way. So what you're seeing here is you're seeing those cubes, which are untrained animals that were run, and this is 24 hours after exercise. The triangular three-dimensional objects are the untrained animals exercised 3 hours after exercise, and then the circles are pre-exercise untrained animals. And the point is, they're all over the place. Okay? You can't tell which animal didn't exercise versus the time point after. And this goes with the story about the compression of the variability. Now, when you made them highly fit, all of a sudden, the times cluster beautifully. The metabolism of the horse, you could now tell the difference between the pre-exercise versus 3 hours later versus 24 hours. And that shows through when you do something called a volcano plot. You then put the data to be able to say, when does it reach a threshold of significance? So in the untrained state on the left, there was only about 30 metabolites that reached that level of significance after exercise. And that's shown here in the blue circle below. So only about 30 metabolites at 3 hours after exercise were different. However, in the trained state, at that same time point, we had about over 100 metabolites that were different. And so you can see that because the response is now clustered, at 3 hours, you got much more significance. You were able to pull out a pattern, a metabolic signature, if you will, for the time. So let me give the highlights. So the number one signature that came out right after exercise in horses had to do with branched-chain amino acid metabolism. So whether or not the horses were trained or untrained, this was a conserved pathway. It was just greater significance in the trained animals because of that ability to have them cluster and be more homogeneous in response. So a normal response in healthy horses, especially very fit horses, is to have high branched chain amino acid catabolites in the skeletal muscle. So they're burning up a lot of branched chains in their post-exercise. And here you can see this visualized a little differently, where we have a number of these breakdown products of the branched chain amino acids. And you can see it in the blue box whisker plots. Those are all the highly trained animals. And in all cases, branched chain amino acid levels, these breakdown products in the muscle, much higher than in the trained state. So this is a static measure, and this is in flux. So it's impossible for me to tell you that this is because whether or not there's a bottleneck and they're getting backed up or they're getting utilized more. Can't tell you that from this data. But we do know that it's a pathway that's very relevant. Then we looked in the blood, and we also see in the blood of horses after exercise, trained horses have higher circulating levels of the branched chain amino acids. They also have a higher circulating level of phenylalanine. So, in the human literature, as well as the rodent literature, there is this concept, this idea of a metabolic signature of type 2 diabetes. It basically is high branched chain amino acids along with large neutral amino acids. And it can be predictive of diabetes by up to a decade. So what we're seeing in the horses looks like this metabolic signature of a really unhealthy, obese, type 2 diabetic, but in fact is a really highly fit horse. We see this in the human literature. Really well-trained humans also have the same kind of signature. The other part of that signature has to do with an accumulation of complex lipid species inside the muscle. So we see this post-exercise in horses and really clearly in the highly fit horses. In the human literature, if you saw this, again, you would say very unfit, insulin-resistant type 2 diabetic. But you also see this signature in really highly trained endurance athletes. So how can we resolve this? Well, there is this concept out there. It's been called the athlete's paradox. Okay, and the idea is that in someone that is an unfit, obese, type 2 diabetic, the reason why you have this accumulation, because there's a bottleneck in the system, and it is not being fed and burned to the mitochondria, and that's why it's accumulating, leads to lipotoxicity, and further making insulin sensitivity worse. Okay, but in a highly trained person or a highly trained horse, in fact, you have mitochondrial expansion. And when you expand the mitochondria, you make them more functioning, you actually need more of these lipid species, these triglycerides, to be able to promote that endurance exercise. So in fact, what we're seeing is essentially, it's a signature of highly trained state. That, horses like humans, I think, use branched chain amino acids, and they need these muscle triglycerides for endurance performance. So I'm going to come back now to the review that I started with. And I think what I've tried to share with you is we've looked acutely, we've looked at these signaling effects, we've looked at mRNA, we definitely see big responses early on, and we think that, they're important, but as we look over time, and once these horses become highly trained, those responses disappear. Instead, what we see is metabolism has changed. We now see that there's a whole new metabolic state that likely has to do with the mitochondrial function of these animals. So with that, hopefully I left a lot of time, so there's questions or anything else. I want to thank you for your attention. I have to thank again my lab and everyone that participated in this work. And I'll do my best to answer any questions you have.